Are you on the call, please?
Yes, I am. Good afternoon to those in Nigeria and good day to everyone. Once again, thank you for joining the call and welcome to the sixth Ibadan Bacteriophage Welcome to the sixth Ibadan Bacteriophage Research Team World Phage Week Webinar for this year 2025. And I want to congratulate all phage enthusiasts and researchers for having Phage Week, and we all acknowledge our contributions and research towards the development of bacteriophage. And as the moderator has stated, this year's Phage Week webinar is centered upon phage therapy, the advancement and impact of phage therapy. And within the next 2 days, we will be gaining more knowledge and insights on the cutting edge work and impact that our research on phages and phage therapy has done in fighting antimicrobial resistance and in clinical applications. So I want us to sit back and relax and enjoy all the sessions that we have brought to you. And to all our speakers and attendees and supporters that have come together to make this webinar lively and to breathe life into it. I want to appreciate you all and thank you very much once again for making this gathering possible. Thank you. Over to you, Koya.
Thank you, Mr. Said Adeduku, for that welcome address. And so the next thing on the agenda is the citation of the first speaker, Dr. Sabrina. And I'm going to be calling on Mr. Warith Ayobami to please take the citation. Thank you.
Good afternoon, everyone. I am Assis Waris Ayobami. I'll be reading the citation of Dr. Sabrina Green. Dr. Sabina Green is a phage therapy researcher dedicated to advancing the clinical use of bacteriophages against multi-drug resistant infections. She co-founded Tailored Antibacterials and Innovative Laboratories for Phage Research at the Baylor College of Medicine, Austin, Texas, a pioneering not-for-profit initiative that sources and prepares therapeutic phages for patients with serious drug-resistant infections. Taylor Antibacterials and Innovative Laboratories for Phage Therapy and its clinical partners have successfully facilitated phage treatment for multiple patients in the United States through the FDA's expanded access and investigational new drug pathway. Currently, Dr. Sabina is a postdoctoral research at KU Leuven, Belgium in the Laboratory of Gene Technology. Beyond her research, she is an active advocate. She's an active advocate for patient access to phage therapy and for uniting the global phage community toward collaborative, patient-focused innovation. Thank you so much.
Thank you very much, Mr. Ayobami, for taking the citation of our first speaker. And on that, we're going to be inviting Dr. Sabrina, our first speaker for today, to please take the stage. Thank you.
Okay, apologies, my moderator. I just received a message from Dr. Sabrina that she's having a little bit of technical issues. So I believe we should be able to if we'll be able to give her some few minutes to sort it out and join the call, please.
All right, great. Thank you. All right, thank you very much, everyone, for staying tuned. Dr. Sabrina is trying to get herself together and join the call. So while we're waiting for the first speaker, I think that we can have the citation of the second speaker so it can take the stage. Um, yeah, that would be Professor Ayman. And um, let me confirm if he's also on the call with us. Yes, good. All right, so I'm going to be calling on Ms. Damilola Bakare to please take the citation of Professor Ayman. Thank you.
Good evening, good afternoon, everyone. I am Bakari Damlola. So I'll be taking the citation of Professor Ayman El Shibni. My apologies. So Professor Ayman El Shibni is a distinguished microbiologist whose research focuses on bacteriophage therapy as an innovative alternative to antibiotics for controlling pathogenic bacteria in humans, animals, and food systems. He earned his PhD in microbiology from the University of Nottingham, UK, and completed multiple postdoctoral fellowships at the University of Nottingham, UK, Cardiff University, UK, and the Evergreen State College, USA. Currently, he works as a professor of biomedical sciences at the Zewel City of Science and Technology, Egypt, where he teaches and supervises courses in microbiology, genetics, and scientific ethics. He's also the founder and director of the Center for Microbiology and Phage Therapy, CMP, and has served as Director General for Research at Zewel City. His pioneering research on phage therapy aims to advance bacteriophages as therapeutic tools to combat antibiotic resistant pathogens. Professor El Shibni is also a certified food safety lead auditor and Food Safety International instructor at iFeed International, with numerous peer-reviewed publications to his credit. Thank you.
Thank you very much, Ms. Damilola, for reading the citation of our speaker, Professor Ayman. Thank you very much for being on the call. You may now begin, sir.
Hello, everyone. How are you? I wonder if Dr. Sabrina is not. She's not ready yet. I am ready. If this is fine with her and with you to start. So
Yeah, yes, I'm here. Sorry.
it is up to you guys now. So will you start?
Yes, if that's okay. I don't think I can go more. I can't take over your spot. I have to go. So it's best if I start now.
Yeah, please.
Okay. Thank you.
Oh, welcome, ma'am. Welcome, ma'am. So sorry for that. So sorry for that.
I'm sorry for my technical delay. Hopefully, I won't have any issues now.
Oh, we really hope so. Thank you very much for coming around. You may start, ma.
Okay, I will share. So can you hear me and see my slides?
Yes, we can.
Okay, great. I just wanted to say thank you for the people who have organized this. I'm very happy to talk again. This is my second time speaking, and so I'm happy that I can talk to you. What's happened since then? So I'm from Texas, and they say everything is bigger in Texas, and it's true. We have a large land, we have big rockets, I'm from Houston, Texas. This is a picture of my daughter. We're standing next to a SpaceX rocket in NASA. And we also like big portion sizes. It's yeah, we like big pieces of meat. And again, I'm from Houston, and we like to say, yeah, we have a problem. Houston, we have a problem. And we definitely have a big problem in Houston and worldwide. Antibiotic resistance, of course. Houston in particular was a hot spot for antibiotic resistance. There's lots of superbugs there. The WHO has referred to this even before
Dr.
the
Sabrina,
COVID.
sorry
Yes,
for the interruption. Your screen is not
yes,
moving, ma.
my screen is not moving. Okay, strange. Is it moving now?
No, ma.
No? Oh, see, maybe in presenter view. Do you see it now?
We can see the screen, but it's not moving at all.
It's not moving. Okay, let me stop sharing and then try again. Entire screen maybe. Try my screen anyway. Whoa, creepy.
Great, we can see it now.
Okay,
Okay.
and it's moving.
Yes, it is.
Yes, yes.
Okay, great. Yeah, okay. So we have a big problem. So the WHO refers this as an invisible pandemic because nobody talks about this. They call this a global epidemic. And the numbers are showing this 1.22 million deaths due to bacterial AMR in 2019. Now the numbers keep updating from 2025 to 2050. They forecast 39.1 million deaths attributable to AMR and 169 million deaths associated with AMR. But the WHO has been also not only talking about the problem, but talking about the potential solution. And they have these webinars now where they discuss about phages to combat AMR. So I come from the U.S. and I was asked to talk a little bit about the regulatory side of phage therapy. So in the U.S., you can get phage therapy through the Expanded Access Program, which is investigational drugs, biologics, or medical devices that have been approved or cleared by the FDA, and the FDA has not found these products to be safe and effective for their specific use. So we applied for these expanded access permissions for patient treatments when I was there through this nonprofit called Taylor. We published on this in this paper in CID, and you can access this paper, and it has everything on there about how we applied for these expanded access permissions. But here's a list where I show you everything that we included when we applied. So basically, you have to show that the phage is doing what it's supposed to be doing, killing the bacteria, and that your preparation is safe for human use. So here's a list, everything from stability testing to doing plate assays, looking at plaques, to doing liquid assays as well. And also, since we purified the phages for IV treatment, how much endotoxin was present, sterility testing, And we included some extra tests as well, looking at antibiotic synergy to be sure that the patient who is going to be treated had antibiotics that the phages that we provided would synergize, and there would not be any antagonism, which can happen. So while I was in the U.S., we received about 50 patient cases. You can see that among these 50 patient case requests, only 12 cases were we treated during that time. So there were very different reasons why. Some patients, they improved over time. It took us so long to sometimes get these phages ready that they sometimes improved. But on the other side of that, sometimes the patient died. And there were times when we could not find a phage, and there were times when we could not get the bacteria. So this is sometimes something people don't talk about as much, is that, yeah, there is a phage for every bacteria, but doesn't mean that you can find that phage. And also, you know, there's patients that have infections where you cannot isolate the bacteria. So how do you treat those patients? And yeah, we couldn't at the time. You can see that these patients had all different types of infections, CF, usually something with prosthesis, UTIs, prostatitis. Among these cases that we treated, they were also different types of infections, from UTI to prosthetic joint infections, to bacteremia, left ventricular assist devices, and wound infections, and from different types of bacteria, from E. coli to Klebsiella to Enterococcus, Staph aureus, and they were all treated with antibiotics, usually IV, and at relatively high doses compared to the doses in Europe. And the patients were also treated at the site of infection if necessary too. So I mentioned earlier that we looked at antibiotic synergy with the phages. So this is called a synogram. And you can see that on the y-axis is the antibiotic, and on the x-axis is the phage, and with increasing concentration. So we wanted to test whether our phages were synergizing with the antibiotics that the patients were going to be on. And so the white here indicates 100% reduction, and the dark orange indicates 0% reduction. So we like to see that as you increase antibiotic and phage, you see white. But there was one case where, yeah, when we were increasing antibiotic and phage, you started to see more dark orange, which suggested possible antagonism. But luckily, we had another antibiotic in hand where we saw synergy. So we were able to treat the patient with a different antibiotic. So what about success? We like to measure this using microbiological outcome and clinical outcome. So microbiological is very definitive. Is there bacteria or not? Clinical outcome is based on the clinician's own opinion. Did the patient get better? Did he feel better? Is he no longer in pain? It can actually vary with the microbiological outcome. So you can see here that we had about 42% favorable microbiological outcome and 58% favorable clinical outcome. So these numbers seem low, but these are some of the worst patient cases, patients that had been on antibiotics for decades. And it really wasn't until we intervened with phage that they started to get better. So these treatments, they can take a while. They can go from 3 months or 4 months to 2 years at the time. And so this is for various reasons. One of them, as depicted in this graph, is that if we can't find a phage, then we have to go looking for a phage out in the environment. And that can be time consuming, or searching for collaborators that have the phage. So you can see that over time, as you start to build your library, that this becomes shorter. But also another hurdle is in the US is this regulatory hurdle. Having to ask for this expanded access at times can delay things. And so this can be an issue sometimes. So among these cases, we had some successes, of course, and they have gone on to talk about their treatments and their success. The first Mayo Clinic cardiology patient that was treated with phage therapy, and then Greg Breed, who received our phages. He had the prostatitis infection, decades-long infection. He hadn't gone without pain for a long time, so we were able to relieve his pain finally. So I spoke to you about our work in the US, but currently I'm in Belgium. And why am I in Belgium? Well, they do many things very well. They have really great fries, really great chocolate and beer. But maybe what you don't know, or you do know, is they have a very nice phage program here. So unlike the US, where you have to go through this expanded access program, they have a different, unique way called the magistral system that they can use. So not just Belgium, but now Portugal uses it as well. So in this system, clinicians can prescribe phages. They prescribe a phage, and then they can produce these phages for personalized treatments. So these phages are in a bank, and then they are produced in a controlled environment. And then they have external quality testing done by a different lab to be sure that the phage, the production strain, and the batch is clear. And then they are delivered to the hospital pharmacies, where the pharmacist then can make these preparations for the patients. So it's very nice. It's straightforward. It's very practical, and I think it's something that I hope that other countries will adopt as well. So we like to say, we like to call it the Phage Valley, because over time, the people here have developed quite a bit of expertise, everything from IP legislation to the clinical implementation of phage therapy of our different universities and different regions around Belgium. And there's a website where we talk more about this called phagevalley.com, where you can read about the history of phages and phage therapy in Belgium specifically, and what's going on now recently in our current research. So currently, all of this has been building up, and it's recently been published in Nature Microbiology. And this was a study led by Dr. Jean-Paul Piernet, and it's 100 consecutive cases of phage therapy. So it's a nice number. Finally, we have a lot of evidence to show the efficacy of phage therapy. And here they saw a clinical improvement in 77.2% of cases, and targeted bacterial eradication in 61.3%. So I will talk just a little bit about some of the work that we did to assist with this 100 case study. So we only came in at the end, or I only came in at the end, but this is data that has been building up over the course of a decade. So what we do is we sequence the strains after patients get phage therapy. And we look at resistance genotypically and phenotypically, resistance to the phage. So in this case, there were 5 cases among the 100 that needed assessment. So this was specifically Pseudomonas aeruginosa. And yeah, we do whole genome sequencing. We do small we do long read and short read sequencing, and we create these nice hybrid assemblies to get a clearer picture of everything that's happening, from the prophages to the plasmids that are present in the bacteria. And then we look at SNPs to see what changes are occurring in the bacteria with resistance to the phage. So this is one patient, patient number 30, that received this phage called PNM, and the isolate was resistant to the phage. And what do you see when there's resistance to this phage? Well, this is a really well characterized phage that uses the type 4 pilus. And when we see resistance, we see mutations within this region. So it makes sense that it's the receptor that is changing. Same with this patient, patient 54, we see changes in the receptor within this type 4 pilus. And what we also like to look at is, are there, with resistance, are there fitness costs? And we do this with the Galeria melanella model. And what we find with patient 30 that had this mutation in PIL-C, we saw no real difference between the mutant and the wild type. So no significant reduction in mortality. Whereas in this patient 54, in this mutation in PIL-R, we do see a slight delay in mortality, but really not much. So even though there's changes happening to this important virulence factor, at least we don't see any major changes in virulence using this assay. So this patient 91, what's often said, maybe in the literature or in talks, is that you have to use a cocktail of phages, because if you use a cocktail of phages, the bacteria won't become resistant. Well, in this case, they did use a cocktail of phages that targeted 3 different receptors. So one phage, PNM, targeted the type 4 pilus. This other phage called 14-1, which is also really well characterized, targeted LPS. And this third phage, PT07, which is less characterized than the other 2, but is highly similar to a phage that uses this MEX OPRM efflux pump as a receptor. Also the bacteria became resistant to. So what happens when it becomes resistant to all 3? You see changes in all 3 of these regions. What we also saw too is a mutation in this gyrase A within this quinolone resistance determining region. So you have a change in an efflux pump, you have a change in gyrase A. Naturally, you have a change in susceptibility to fluoroquinolones. And in this case, for sure, the patient's isolate became resistant, but then had resensitivity to ciprofloxacin and levofloxacin. So again, we looked at fitness defects using the Galeria melanella model. So in this case, when you have mutations in all 3 of these regions, you see that the Galeria, they survive this infection. So there are within multiple mutations in this regions are associated with lower virulence. So again, resistance can be good. You can have resistance and then have resensitivity to antibiotics. You can have resistance and lower virulence. And finally, with these two patient cases, what we see is that when you have resistance, it's because you isolate a completely different bacteria. So that's kind of what happens with phage. It's so specific that naturally, yeah, maybe you become infected with a different Pseudomonas aeruginosa that the phage can no longer or cannot infect in the first place. And that's why I believe it's important to have antibiotics as well to take care of these other types of infections. So both patients, even though they were infected by another bacteria, they clinically improved. So yeah, and why did they clinically improve is because phage and antibiotic, I think, are stronger together. And I've highlighted two studies that I was involved with that kind of shows this, is that with phages and antibiotic in patients, I think is much better than alone. So I'm just going to talk a little bit about this project that we have, thinking about the global picture, not just what's happening in the US and in Belgium, but also what's happening in Ukraine. This is a collaborative project to combat multi-drug resistant bacteria in war zones. So the AMR crisis is already really bad, but it's only made worse by war. Of course, you have severe injuries. You have antibiotic overuse. You have clinicians that are, trying to treat the patients as best they can, but this can happen. And you have displacement of people around the world. They may not be treated as well where they are, and so they have to go to other hospitals in other countries. So naturally, this is a recipe for the spreading of AMR. So we started this phage support. Oh, sorry, what happened? Okay, sorry. Okay. So we started this Phage Support Ukraine initiative, and we're working with a hospital in Kyiv and a military hospital in Kyiv. They send us bacteria isolates, problem isolates from their country, as well as hospital waste. So this allows us to find the phages. As we know, the hospital waste has the bacteria there, and so the phages should also be there. My work was mainly focused at looking at the strains that are currently there and characterizing them and characterizing the phages that were isolated at the Queen Astrid Military Hospital. What we realized is that the people there, they may, even though we could send them phages, they're not quite equipped to keep the phages and to work with the phages. So laboratory equipment and consumables were also sent there. And the phages were isolated, and they will soon be sent there when they have everything set up so that they can work with the phages. So the hope is that the people there will be able to treat the patients that are in Ukraine.
Quiet. That's only Lily. Come on, quiet.
So among the strains that we received, so they arrived in these filter papers. Klebsiella pneumoniae was the bug that we saw the most, and it was the most antibiotic resistant. You can see this is an antibiogram of what we typically see. Lots of RRRs, resistant to everything, except at the time was sensitive to colistin. We performed a whole genome sequencing on these strains, and we see that they group within these 4 major sequence types. But there were 3 isolates highlighted in yellow that kind of were a bit different from the others. Looking at what they were, we thought they were ST23, but when we look at all the currently available ST23 genomes, there were some that were a bit different from the rest. And so if you take these ones that are a bit different and you kind of focus on them, and instead of doing MLST, you do CG-MLST. So instead of looking at a few genes, you look at hundreds of genes to better characterize these bacteria. You can see that this is all one clonal group. So this is CG10146. And when we look at the metadata of where these come from and when they were isolated, you can see that many of them were isolated recently. They come from different countries, from the US to the Netherlands to Denmark, Germany, and Russia and Ukraine. And they have high virulence scores. So Klebsiella pneumonia is sometimes ranked from 0 to 5 for how many virulence genes are present. And so they have high virulence scores, and they have high resistance scores. So looking further into the metadata, we could see that many of them actually have Ukrainian origin. So these are patients that were displaced due to the war in these hospitals around the world, but they seem to be carrying the same clonal group. You can see that there are a few that are a bit different, though. I highlighted them with these red arrows. They were isolated much earlier. They tended to have high virulence scores, but low resistance scores. And you can see that the origin is actually from Russia. So it seems like from this data that we have this clonal group that may have started in the beginning of this conflict, possibly in this region, and is now beginning to spread due to the war. So this is kind of it's a story that really highlights what conflicts can do in this situation. We've also characterized strains from patients that were displaced in Belgium and Latvia and Germany. And again, you can see that they fall within this group, this 10146, but also many of them fall with this other major sequence type, ST395. They carry lots of resistance, they carry lots of virulence. And how do they carry them? Well, within these huge virulence plasmids. This is an example of one. It's 364 kb. This one in particular is carrying arabactin, the siderophore. It carries RMPA. This is a regulator of mucoid phenotype.
Is it me or I can no longer hear you?
I can also hear her. I think it's from her
Okay.
hand.
Dr. Sabrina, we can no longer hear you.
I can't hear you.
No, we can't hear you, ma. Oh,
Can you hear me?
oh yes, we can now.
Okay, I'm so sorry for all these issues. Okay, I will try to go quickly. So can you see the screen still?
Yes, ma.
Okay, so now the situation has gotten worse. We see lots of resistance. They're still falling within this sequence type. So talking about phages just for a couple minutes, they were able to isolate several phages against these strains. This is an EOP chart. So yeah, we have phages against all these strains. One particular phage, this was named Mavka. We looked at resistance. This is in vitro, and we saw an interesting phenotype. So in 3 out of 9 times, we saw complete loss of this plasmid. So these strains, they become resistant, but they lose this huge virulence plasmid. So again, yeah, this is something that we like to see, that, yeah, if we give this phage to, for instance, we could then see sensitivity to antibiotics. We can see reduced virulence. And we see that when we look at the antibiogram of these strains, it used to be RRRR with only sensitivity to colistin. And now we see sensitivity to aminoglycosides and this antifolate sulfonamide. So yeah, again, resistance can be a good thing. So yeah, thinking about if you're thinking of a strategy to kill something as dangerous as Klebsiella pneumoniae, yeah, you look to where they keep all their weapons and their shields. In this case, this plasmid, right? And you use phage to specifically target it so that you can remove all its weapons and its shields, and you can really get rid of it. So I'm really sorry for all the issues I had, technical problems, but I'm happy to take some questions.
Thank you very much, Dr. Sabrina, for that eye-opening, enlightening presentation. We really appreciate it. Yeah, for me, it's so sad to see how war is contributing to the spread of antimicrobial resistance, and the fact that, well, sometimes these resistance can be a good thing in discovering and in innovating. Thank you very much for that beautiful presentation. We will now be giving chance for you to ask your questions. You can ask all your questions now. Um, if you have questions, please raise your hand so that I can know the number of people that have questions. Thank you. Uh, I know that some people have joined us through the YouTube channel. If they have questions too, can we know? Sahid, you have a question? Please go ahead.
Okay, thank you. Okay, thank you very much, Dr. Sabrina, for the excellent presentation. Um, so I have a question. Now, as regards the regulatory burden of phage therapy, I want to ask, what are your recommendations whereby low and middle income countries that also face this AMR burden can also get easy access to this phage therapy, these phages for their treatment of antimicrobial resistance?
Yeah, so you're asking how low and middle income countries can get access to phage therapy, correct?
Yes, ma'am.
Yes. Yeah, I see it the same way that we are dealing with Ukraine. You know, I think if I think your countries should be able to produce your own phages, as long as you produce them safely, and it doesn't cost a lot. It's really it's just a regulatory issue. So you need to be talking to your regulators, and to discuss with them what's happening in countries in Europe and in the US, and why can't your country do it as well? I believe you have the facilities to do this, and I think why not? Of course, also we can provide phages too, and then you would just have to administer them yourselves. But it's not something that requires high income facilities. It's really it can be done in regular labs, as long as you meet all the safety requirements before you administer the phage to the patient.
Thank you very much, Dr. Sabrina, for that answer. We have another person. Please go ahead with your question, Kupatsa. Or please go ahead with your question. You're raising your hand, Kupatsa. All right. If there are also questions from the YouTube channel, please just drop your questions in the chat space or on the comment section at the
Hello?
on the YouTube page. Yes, please go ahead with your question, Kupatsa.
Yeah, sorry, my microphone wasn't working. I'm new to this phage therapy thing, but my question is, but I'm interested. But my question is, upon administration of the phages to the patient, how long do we expect the phage to last within the bloodstream? And is there any possibility that the phage does undergo mutations and becomes a human or eukaryote cell targeting species?
So yeah, interesting questions. So the first question, I think it's very quickly, your system, depending on you administer it, but IV, your body is filtering your blood, and so it quickly like gets rid of it. But phage are pretty quick acting, so then once they infect the bacteria, then they will start replicating, right? So the point is to really give doses to allow your phage to replicate. So if you give too high doses, then it just kills the bacteria. But you want to give them these lower doses so that you have an active infection on the bacteria. And of course, then if your system eliminates it all, at least that your phage are now replicating in the bacteria. But I can't give you exact numbers. It's something that is being worked out by other people, and it may depend how you administer it, what phage, what dose, what type of infection, these types of things. The second question, I can for sure answer that one. Phage have been around for a long time, long before us, and they have yet to have evolved to infect a eukaryotic cell. I think it's very would require a complex evolutionary event for this to occur, and so I for sure don't think a phage can actually infect. So infect, replicate, kill the way that it does a bacteria cell. Maybe it can get inside a eukaryotic cell just because they're very small, but or attached to a eukaryotic cell, yeah, sure, but infecting it the way that it infects bacteria, I don't think that's going to happen.
Thank you very much, Dr. Sabrina. I hope that answers your question. Um, yeah, we have another question in the chat space. Says, can you please describe a bit about how we can isolate and characterize phage?
How you can isolate and characterize phage. Yeah, you can go out in the environment. There's lots of protocols. There's a video I made actually of how I isolated phages, but it depends on what phage you want to isolate. We turn to sewage. We go where the bacteria are. Then it's simply incubating it with the bacteria, kind of making the phage in there increase, and then it's just filtration and isolating your single phage. It's not a complex thing. Many students do this in universities. I think anybody can do this. What's more complex though is yeah, the further characterizing, as you said. You can isolate it, but how you characterize it, sequencing, for instance, doing different assays to see how well it infects the bacteria. And then when you're thinking about patient cases, there's probably separate criteria for a good phage for patient treatment. But I think anybody can isolate it, and I think you can just look at protocols online, and there's nice protocols for learning the basics about phage isolation and characterization.
Thank you so much, Dr. Sabrina, for that excellent answer. And also to all the answers you have provided to all the questions. We are so delighted to have you here, and we really appreciate your presentation. And you, thank you very much for educating us.
Yeah, thank you for
Um,
having me. Thank you.
yeah. So we're going to be having a display from our partner for 2025 edition of the World Phage Week Webinar series. That's Norgreen Biotech. I hope that they're ready.
Good evening.
Good evening, sir.
Yeah, thank you very much for organizing the event. This is Dr. Ayman Shibini, Professor of Biomedical Sciences and the Director of Center for and Face Therapy at the City of Science and Technology. I would like to start with thanking the IPERT organizers for inviting me to present in the Phage week in 2025. My presentation title is about next generation phage therapy, shaping the future of antimicrobial resistance. Just turn the video off so I can focus with you guys. So, can you see my screen now?
Yes.
Yes, okay. It is my pleasure to present the Center for Microbiology and Phage Therapy and present my research team's work over almost more than 10 years now in Egypt. So simply, I will take you in a journey to display why we are interested in phage therapy. And the main answer is due to the antibiotic resistance and the multi-drug resistance, which we can see worldwide right now. So in general, I will go through the problem in a few minutes and highlighted why we have a AMR and superbugs worldwide. This is because of using antibiotics in different areas, especially in medical area, either outpatient or inpatient, in addition to use in addition to the use of antibiotics in agriculture as a growth promoter. So all those contribute to the multi-drug resistance due to the misuse or abuse of using those antibiotics. Nowadays, we have to highlight something as well, that when you use antibiotics, you destroy and damage and get rid of all or most beneficial bacteria, which support us in different ways. Our body has almost 10 times more normal microbiota compared to our cells. So we have 10 times normal microbiota, and all of them are beneficial bacteria, which support our body by digesting the food, providing and making different vitamins like vitamin B and K, control and compete with pathogenic bacteria by competitive exclusion, protect our GIT from those pathogenic bacteria. And due to those factors, normal microbiota is considered part of our innate immune system. So taking antibiotics to control or to kill pathogenic bacteria, unfortunately, start killing those normal microbiota, which are highlighted in yellow color here. And by the time, once we kill all or most of normal microbiota, the pathogenic bacteria, which enrich circles, started to propagate, increase in number, and take over and cause inflammation and other, maybe end up with serious diseases or death to human body. So the anti-resistance worldwide is a big issue nowadays, and the World Health Organization is expecting that the number will be 10, with the number of people who will die due to AMR by 2050 will be around 10 million person. And for sure, this number is not valid anymore. Once Wellcome Trust did many studies, and they expect that the number of people who will die between 2025 and 2050 will be more than 39 million people. And this number is almost near to the reality due to increasing number of people who die due to antibiotic resistant bacteria. So all people now, including the World Health Organization, FDA, and all health organizations, to find alternatives to antibiotics. What different alternatives that can be used to treat human infection and animal infection instead of using antibiotics? As you know, there are many alternatives. Some of them are in the market right now, like vaccination, probiotics, botanical plants, antimicrobial peptide, and bacteriophage therapy. So we take the advantage of all those alternatives to develop new therapy, new delivery system, find the synergistic effect of using bacteriophages to control pathogenic bacteria. And I will share with you some of those results, which we already published most of them. So for people who don't know what is phage therapy is using bacteriophage, which is a virus that can kill only bacterial cells to control those pathogenic bacteria without affecting, hopefully, the normal microbiota. The bacteriophage has two different cycles. The lytic cycle, which is used by virulent phages to produce a lot of phages that can kill other pathogenic bacteria, as we can see in this video in the left-hand side. And the other cycle, which is lysogenic cycle, is used by temperate or prophage. And simply, we can differentiate between those two different group of phages by doing whole genome sequencing, and in addition to other phenotypic characterizations that can be identified based on our experience. So what we do, we start. I hear a question about how to isolate phages. Simply, I will go through the main steps which everyone in phage therapy use to isolate, identify, amplify, characterize phages, and share with you some of the outcomes of different applications and experiments that have been conducted in different research area, either in clinical research or in food, agriculture, or in vet. So simply we start with phage isolation and characterization. Some of these characterizations, as you can see, the left-hand side, those are some phages which are specific to Acinetobacter. We do find the right phage and we do isolate it and do purification. Then we do amplification and do different characterization like imaging by using transmission electron microscope. And then we do this phage amplification. Here we are using a bioreactor. As you can see in this bioreactor or fermenter image, on the right, on the left-hand side, it is highly turbid, and this was done at the beginning of the experiment when we inoculate the broth with the bacteria. Then we infect the bacterial culture at certain OD with the phage, and after amplification, we can see the broth on the right hand side here of the in this figure that it is almost clear, which due to the amplification of phage. And here we can see the increasing in number over time for two different phages due to the amplification process. Then before we do other or more or further experiments, we need to study the phage stability based on their application. So I take you in a journey from phage isolation to preclinical applications. So for phage stability, we usually, for example, if we use it in vet, we need to study the stability of isolated phages to temperature, to UV light, and to pH according to the application. So in case if we apply it in poultries, then we need to find how phage can tolerate the pH of the stomach of a chicken or large animal or whatever is the application. Then we do what we call phage cytotoxicity if we apply it in human or in animals, and do the genomic analysis to identify if the phage sequence has any lysogenic resistance or any things that inhibit or control its use in further therapy application. So I will share with you some of our results now for phage isolation and application in different on different clinical isolates from different diseases. The first one is lung infection. So we did a lot of work. I think I can hear that there are some noise. If you don't mind, to close the mic, please. So I will start with some of our good results, which we published already by one of my PhD students in 2023. And this study was investigating the isolation of bacteriophages against Klebsiella pneumoniae. And study, we already isolated very good phages, and we did the characterization of those isolated phages. And this phage, which is ZCKP20P, is the candidate for this study. When we applied the phage, we usually study what we call multiplicity of infection, which means how many phages we need to use against how many bacteria. So multiplicity of infection 0.1 means that one bacteriophage against 10 bacteria, MOI1, which means one bacteriophage against one bacteria, MOI 10, which means 10 bacteriophages against one bacteria. By doing by studying the killing kinetics of and the interaction of phage with bacteria, we identify the most effective multiplicity of infection. In this case, we can find that multiplicity of infection 1 give us the highest results. Here is the control in color. The oh, excuse me. Can you please turn the mic off?
Okay, I'll do that.
There is a mic, open mic, and you can hear the noise. If you don't mind, the guy who opens the mic to close it, please. Thank you.
Thank you, sir.
Oh. Hello?
Yeah, please go ahead, sir. He has muted his mic.
Okay, thank you. So in MOI1, which is the middle figure here, we can see that the bacterial control is blue color. This is the curve of the control, and the green color is bacteria after treatment over time. So, and this is the red curve is PFU, which is the bacteriophage titer after infection. And here we can see that we have almost about more than 4.5 log reduction, which means that we get rid of a lot of bacteria during this in vitro treatment. Also, we studied how this phage can inhibit the formation of biofilm and also to clear the already produced biofilm, which we call it biofilm clearance or degradation. So in the left-hand side, we can see that this is the control, untreated one, and those are the treatment with different multiplicity of infection, and there is a big and significant reductions in the number, which means that the bacteriophage was able to inhibit the formation of biofilm. On the right-hand side, which is figure B, we can see also that this is the untreated one, and this is the biofilm, the formed biofilm after the treatment of with bacteriophage. We see that the phage was able also to eradicate and degrade the formed biofilm in a significant way, which means that the phage can control the formation and clear the formation of biofilm in a significant way. And this, of course, is an important advantage compared to the antibiotics, because you know that the biofilm forming bacteria can resist antibiotics up to 1,000 time compared to the vegetative cell. One more study, but here with Acinetobacter. Again, this is Acinetobacter can cause pneumonia as well. And if you can see here the killing kinetics of phage RMA1, it gives us a significant reduction in the OD of bacteria at different MOIs compared to the control. And you can see this is clear in the wells of the 9-6 wells plates. This is the control one, and this is the eradication with using different multiplicity of infection. And here we can see on figure B, this is the percentage of reduction of bacteria. Untreated, there is almost zero reduction. The bacteria can grow very well without any reduction, while when we used different multiplicity of infection, we see significant reduction, almost around 95% or more reduction compared to the control. Also, when we studied the same phage to see its ability to inhibit the biofilm formation of Acinetobacter and degradation, we can see this is a control, the untreated one, for the inhibition, and those are the different multiplicity of infection, and the OD was very low compared to the control in biofilm inhibition. And here for with phage biofilm degradation, we can see also significant reduction in the OD compared to the control. So since this phage is isolated to target pathogenic bacteria in lung infection, so how can or what is the safety of using these phages to target those pathogenic bacteria in human? To answer this question, we have to do what we call it cytotoxicity effect, and simply what we do is to use the phage against to control bacterial infection in cell line. And this cell line here is HSF cell line. And in figure A, as you can see, this is a viable bacterial count. The red curve is the viable bacterial cell alone without any phage treatment in the cell line, while the other curves represent the using the phages at different multiplicity of infection, and at multiplicity of infection 100 we couldn't find any bacterial cell after 0 time, while with other MOIs below low MOIs we can see that yes, at the beginning there was a bacteria, but over time it decreased significantly compared to the control, and this is clear here in the viability of cells. If you look at the viability of HSF cells here, this is the control, the cells without any treatment, and next to it the treatment of those cells with bacteria alone. And as you can see, there is a very big reduction in the viability of cells, almost 70% compared to the control, while when we used phages at different multiplicity of infection, we didn't see any significant reduction compared to the control, which means that the phage managed is safe to be used with cell line. And here again, this is the phage titer over time. We saw that the phage was stable. However, it managed to increase in times by replicating on its bacterial host in the cell line. Those are the images of cell lines after treatment with phages. And in the figure number 2 represent low density of cells, which means that the cells cell viability was very low. And when we identified this slide, it is related to the cell lines which was treated with bacteria alone without phage, which again confirm our observation that the bacteria can kill the cell line while the bacteriophage can save it. Another application is to isolate phage and study their ability to control bacterial infection, but this time in urinary tract infection. So we published some good results in this area by isolating specific phages against Escherichia coli. And this some of those results, my student managed to isolate a good phage. And of course, again, we study the killing or replication dynamics of phage by using different multiplicity of infection. And again, here we see that MOI1 gave the most powerful effect in reducing the bacterial number. But if you can see in this figure here, after 120 minutes, we started to show some bacterial resistance, and I will tell you what was our plan to control this bacterial resistance. Simply, we use antibiotics with bacteriophage to study their synergistic effect to control bacterial infection. So we did antibiotic sensitivity assay to identify which antibiotic can affect or kill this bacteria. And we determine the MIC using the plate reader on the, as it is clear in the methodology on the left-hand side. So simply we use the gentamicin, and we identified the MIC of gentamicin that it was around the 500 microgram per liter. And we tested the phage alone. We tested the phage with antibiotic at its MIC, half MIC, different multiplicity of infection, different combination. To make the story short, we identified about 4 different combination of phages with different MOI and with antibiotics at different concentration, and we found that those combination managed to kill completely eradicate the bacteria in vitro. So after that, we did this synergistic effect study, the combination in TSB by counting the number of bacteria and the number of bacteriophage. So as you can see here in figure A, this is the bacterial control, the black curve, and the green one is gentamicin, half MIC. The blue curve is phage alone at 100 MOI, and the purple one is the combination of phage with gentamicin. You can see here that we saw resistance when we use the antibiotics alone. After 4 hours, again, we saw a similar pattern with using phage alone, but when we use the combination of phage with antibiotics, we didn't see any resistant bacteria over time. And when we enumerate the bacteriophage, we saw that the bacteriophage alone without antibiotic increase in number compared to the phage titer when it is combined with antibiotics. Which means that there are other factors that reduce the replication dynamic of this phage. We had to test this before we go the in vivo studies in a rat model. We had to study this in natural urine. So we studied it in artificial urine and the natural urine after getting the IRB approval. And as you can see in the left-hand side again, we can see that the combination, which is the purple color, managed to eradicate the bacteria in urine, but after longer time due to the composition of the urine, which reduce the ability of fish to kill the bacteria quick compared to the TCB. And again, here we didn't see this high replication of phages when we did the infection in natural urine due to the same condition. So we decided to do the in vivo studies by studying the effect of those phages to control E. coli and UTI infection in rat model. And simply again, here's the black color is the control, which bacteria without phage, bacteria with gentamicin in green, bacteria with phage alone in red, and in combination in blue. And again, here we see different results. So the bacteriophage managed to control and reduce the number of bacteria alone, or quite similar to the bacteriophage with gentamicin, which means that the normal microbiota of the stomach of the rat enhanced the ability of fish to control the E. coli infection compared to the in vitro studies. So what is the safety of those phages then to treat UTI infection? We had to answer this question by doing the cytotoxicity and to make it simple. So you can see that the viability of cells is quite similar with different multiplicity of infection using 2 different cell lines in this study, which is HSF cell line and T24 cells, and both of them showed safety and no significant or any impact of using phages on those cell lines. Again, we did similar studies to control UTI infection, but in this time with Pseudomonas infection. And in this paper, Amira did a very good experiment. So we study how phage can control the bacterial infection in urinary catheter. So simply we inoculated the catheter with bacteria, and as you know, Pseudomonas can produce biofilm very, very easily. So in the top figure A1, B1, those are imaging images for the biofilm formed by bacteria on the casseters, and the blue figures represent the treatment when we treated those infection or this bacterial or biofilm formation by using our isolated phage Pseudomonas ZCPS 13 phage, which managed to eradicate a huge number of the biofilm formed bacteria, which means it is very, very effective to control it. And this was clear in the bottom figure in biofilm inhibition here and the biofilm clearance, where we found a significant reduction. More success studies, and this time to control bacterial infection in eye. So simply we publish a very nice piece of work in controlled released journal, and we submitted a patent file of using hydrogel system with phage to control bacterial infection in eye. Simply, this is the flow diagram of our methodology. We made in situ hydrogel, and we embedded bacteriophage in this hydrogel, and we treated rabbit model infected with Pseudomonas aeruginosa. And I share with you now some of those results. Here is the replication dynamic of and killing dynamics of using this phage, which is VBPA ZCPBS1, to control bacterial infection. This is the control, which is the black one, and you can see here a huge reduction, especially with higher multiplicity of infection, to control and eradicate the bacterial count over time. And here you can see the percentage of reduction in the in figure B. Here, this is the untreated bacteria, almost zero reduction, where we managed to eradicate the number of phage up to almost 95%, which is huge. And this is clear in the wells of the 96 source plates. Here you can see that this is the control, and this is low MOI. Then we have higher MOI that managed to eradicate and kill all or most pathogenic Pseudomonas bacteria. Here, this is we had to do what we call phage release experiment, and we to test how phage will manage to be released from the hydrogel over time. And we did this experiment for 24 hours. And as you can see here, once you have the gel and you have the bacteriophage embedded inside the gel, over time, this is a sustained release of phage over time, and the maximum almost approach after 4 hours. And then we have like a stable release of bacteriophage, which means that when you use those hydrogel to treat eye infection, you will have a long time treatment compared to the normal traditional treatment. And here in the figure below, we had to test the irritation of using this hydrogel, and as you can as it is clear that the normal eye has quite or similar appearance with no irritation at all with eyes treated with those hydrogel. And then we had to apply this in vivo by infecting a rabbit model with Pseudomonas bacteria, which is this panel, positive control, and we had to use a negative control, which rabbits without any treatment. And here in the panel 3, the phage in gel, and this is the antibiotic alone, and this antibiotic was effective against Pseudomonas. And when we finished the data, we found that the phage managed to kill the bacteria quite similar to the negative control and antibiotics, while the positive control has opacification. Not only this, we had to do histopathological examination to study how phage can affect the cells of the cornea, and we found that all treatment manage to have normal cells without any side effect on the epithelial cells or whatever. While in the lower panel, we have like thickened or increased the corneal thickness with focal erosion and inflammation when the bacteria alone infected the cornea of the eye. More studies to study the impact of phage to control bacterial infection in tooth. See, all of you know that, or all of us go through this bad experience by having dental caries. So my student managed to isolate a bacteriophage that can control Enterococcus faecalis in a very nice tooth model. So simply we isolated specific phage to this Enterococcus, and we studied the synergistic effect of using this phage with natural product like propolis, and we applied this to do irrigation and medication. And I will share with you some of those nice results right now. This is a root canal of our tooth. So we used a tooth model to study the irrigation and medication. And let us see the results here after the treatment in this figure. This is a control, the bacteria without any phage. This is the normal saline which they use in the dentist use. This is sodium hypochlorite, propolis alone, phage alone, and this is a combination of propolis with phage. And as you can see, there is a very significant reduction in number when you used phage alone, but a better results when we used phage with propolis compared to the control. And when we did some imaging analysis, we found out that the phage managed to save the root canal. This is a negative control without a healthy tooth, and those one which treated with bacteriophage, you can see the root canal are clean and good, and compared to the others. And here is the slides, which in green is the bacteria alone, which means that live biofilm or live bacteria formed biofilm, while the green, the red color means that those are killed bacteria without biofilm formation. So this was a great confirmation to our study, and we did the same thing for medication as well, and we noticed similar results, but for medication, which means that we treat the tooth for 7 days compared to irrigation, which we treat the tooth only for 5 minutes. So again, they use boloxamar for medication in dental clinic, and here you can see dead bacteria in control. The dead bacteria, percentage of dead bacteria was very low, while when we use phage and propolis, almost 80% of bacteria was killed by using phage and the propolis. Again, this is the log number of bacteria. This is the control compared to the treatment. There are almost about 6 or more log reduction of bacterial infection. Same thing for the imaging. I try to go quickly in the first few minutes to share with you some of our good results as well. So we didn't focus only on using bacteriophage. So you know that there are some other phage-based product like protein or peptides or any good product that can be used to control bacterial infection. So we focus on the production of endolysin as well. So to use the endolysin alone. So my student Fatma managed to isolate and clone to identify, clone, and express the first protein and purify it for future application. And this work has been published in Viruses a few years ago. And as you can see here, this is our first endolysin, which we call it lysozyme ZC1. And here you can see the treatment of bacteria Bacillus cereus and Staphylococcus aureus with this lysozyme. Here is the lysozyme itself in black color. This column shows that almost 98% reduction in bacterial count has been approached when we applied lysozyme with Bacillus cereus. And again, with this is 98% with commercial lysozyme. And here the green one is lysozyme ZC1, which is almost around 80 or 80 something reduction in bacterial count. While with Staphylococcus aureus, the percentage of reduction was quite low. And when we tried this enzyme with editor to control gram-negative bacteria, because as you know, most of gram-negative bacteria are resistant to the lysozyme due to the outer membrane of gram-negative bacteria. But with using low concentration of editor, we managed to control the bacterial number. And this is clear in those black column compared to the lysozyme alone, editor alone, and control, which is stress buffer without any lysozyme. Here, as you can see, there are some figures that show the lysis of bacteria. This is lysozyme ZC1 against Bacillus. You can see the clear zone in this Petri dish. It is very clear, like the inhibition zone of phage or antibiotics. And we did some images, and as you can see, this is a control without any lysozyme treatment. And this is the bacterial cell treated with lysozyme. And you can see the rupture of cell wall is very, very clear and nice, which indicates that the lysozyme managed to rupture the bacterial cell wall. Not only this, we isolated a new, identified a new protein as well with a small size, and we predicted the 3D cell structure of it. Again, we tested it against different bacteria. This is one example of one bacteria, which is Micrococcus. We tested against the acne, we tested against Acinetobacter, and as you can see, this is the activity here. This is the turbid Eppendorf, means that the bacterial growth, this is a control with bacterial growth, while the clear Eppendorf in both figures, the treated bacterial culture with this protein. So we had to test this, the synergistic effect of this protein with another, with editor. And here again, editor alone, lysozyme was edited at different concentration, untreated, and lysozyme alone. And again, we saw huge impact of using this new lysozyme with editor, and it is almost 90% reduction in the OD of bacterial culture compared to the control, which means that this lysozyme has a great effect and ability to control bacterial infection. After that, we build on those results, and we decided to take some images to reflect and confirm those results. And we did use the fluorescent microscope, scanning electron microscope, to see with two different dyes, DAPI and PI staining. And this blue, or the top picture in BBS, those are the live cells without any treatment, and at the same time there is no dead cells. And when we applied lysozyme ZC2 on the bottom figures on the left hand side, we can see only few bacterial cells still alive compared to the top one. So the top one is considered a control, and this is a treatment. And again, this is a control with negative, no dead cells. But when we treated, we can see the condensed red color, which means that all bacteria were killed, maybe with only few unseen live cells. So which means that the lysozyme 2 managed to kill its bacterial host. Not only this, again, as I mentioned at the beginning, we develop phage cocktail, we isolate phages to target pathogenic bacteria in animals. So in animals, we have to consider the acidity of the stomach. So we developed a new delivery system, and we build on this model to develop new delivery system as well by encapsulating bacteriophage to manage to tolerate the acidity of the stomach and to allow the sustained release of bacteriophage from those beads. So those are the figures of our beads in lab synthesized or capsules or beads that have been produced, and simply what we do is to embed the phage inside those beads, and they are like gel to be introduced to poultry. And in this figure here, you can see that this is the sterile release over time. We did this experiment over 6 hours, and we see that the phages start to release after 1 hour, 2 hours, 3 hours, up to 6 hours. There is a good release and good stability of phages. And when we treated the bacteria with those beads, we see at the beginning, in comparison to the phage alone, this is the phage alone, which is in the red box. You can see that the phage at the beginning, after 3 hours, give a huge reduction. Then the reduction decrease, then decrease up to 10 hours. In comparison to the beads, low release, then increase release, and the maximum release was obtained after 10 hours. And we need this sustained release when we treat animals or poultry to give the phage the ability to be stable until it reach it's the site of infection. Again, we try to see those the ability of phages to save eggs. As you know, the ABIC, which is avian pathogenic Escherichia coli, can kill the embryo, the fertilized egg. If we don't try to save it, the embryo will die. So simply we isolated 2 different phages, and we make phage cocktail, and we infected this live or fertilized eggs with bacteria, Escherichia coli, ABIC strain, and we incubated it. Then we treated it with a combination of phage B1 and phage F1 and F5, and one treatment with B1 and phage F1, and another treatment was only phage B1. And as you can see, the combination of the 3 phages managed to save the life embryo up to 90%, which means that phages can save the fertilized eggs from infection with pathogenic avian pathogenic Escherichia coli. And finally, 2 more application, and I will finish. So we managed to again to control salmonella infection in milk as well. And as you can see here, this is the control, and those are the treatment with the different multiplicity of infection in vitro, in TCB, and on the right-hand side, this is the treatment in milk. And as you can see, with high MOI, 10,000 and 100,000 MOI, we managed to eradicate the bacterial, the salmonella bacteria in milk. And this is a huge achievement, because you know usually phage cannot easy control the bacterial infection in milk due to the high lipid content. We did some some studies in bacteria like Erwinia amylovora, which infect beers. So again, we managed to control the bacterial infection in agriculture by using bacteriophage to control the Erwinia infection. And as you can see, this is a positive control in the top. This is a negative control. We scratched the pear. And here in the blue, this is the treatment with different MOI. We scratched the pear, inoculated with Erwinia, then treated it with phage. And as you can see, they are quite similar to the control. And this work is under publication right now. Can we use phages to detect pathogenic bacteria, to develop a tool that can detect the bacterial infection? The answer is yes. We published and submitted a patent in this area, and this work all the work I presented are conducted in my research center by my research team. So again, this is a millimeter wave technique in a collaboration with a colleague in nano science, nano electronics. So we developed a tool that can predict the interaction or the replication dynamic of phage in the presence in its hosts, and based on this variation in the waves, we can tell that the bacteriophage can find its host and infect it. Again, we developed another biosensor like lab on a chip. So simply again, we use the phage and its bacterial host on a chip, specific biosensor to study the interaction between both of them and give us signals that the phage can infect this bacterial host and give us an indication that it can identify if there is an infection or not. And this again is submitted for patent. That's it. And I would like to thank my research team. Some of them are not included in their results are not included in this presentation, but because of the time, I couldn't include all the center is research outcomes. And I will be more than happy to answer any of your questions. Thank you very much.
Yeah, thank you very much, Professor Ayman, for that rich, power-packed presentation, featuring real-world applications of phage therapy across board. That was a really enlightening and beautiful presentation. Thank you very much for this, sir. I believe that we have questions that we want to ask, because this is a very beautifully packed presentation. So before we ask our question, for just 1 minute, 1 minute, 1 minute, we're going to be having a quick short video from our partner, um, Novo Biotech. You can also drop your questions in the chat space. Also, we're going to have this beautiful 1 minute presentation from our partner. Now it's about phage isolation, because I heard our speaker talk about isolating phages, which is a very beautiful and important um, process. So this presentation is going to be about phage isolation, and we will present for 1 minute before you ask your questions, and our speaker is able to answer your questions. Thank you very much.
You're welcome.
Yeah, thank you very much, Norgin Biotech. But we couldn't hear anything anyway. So right now we're going to be having the questions from you all to our beautiful speaker, Professor Ayman. Um, if you have questions, please raise your hands. Thank you. All right. We have some questions in the chat space, sir. One of those questions is, thank you so much, sir, for this presentation. Um, can I get to know more about the application of phage and lysozyme?
Okay, yeah. Actually, we have some. We did. I see another question as well. So I will because related to this question, we did some application for on wound infection using phages, and we also now to develop a new delivery system to use phage and endolysin hydrogel to treat bacterial infection, skin and wound infection and skin infection. I mentioned that we tried those endolysin, lysozyme 2, the second one, which we haven't published yet, on acne, the bacteria that cause skin infection, especially in face. And we develop a new system to delivery system to use this endolysin and in combination with phage and in combination with antibiotic to treat those bacterial infection in skin. And we published 2 papers before in wound infection treatment using bacteriophages. Those were conducted 2 years ago, but due to the time limitation, as I mentioned, I couldn't include all the results of our lab. There is another ongoing research to use bacteriophage with another natural with another natural product with antibacterial activity to control Pseudomonas infection in skin as well. And yes, we have other application like we develop a new packaging system. One of my master student is doing this right now to develop a new packaging system, including bacteriophage to control bacterial infection in food. So there are many applications. Some of them were already published, but not included in this presentation, and some of them ongoing right now. I hope I answer your question.
Thank you very much, sir. Thank you very much, sir, for answering that question. I think we also have someone else raising their hand. Kennedy, are you on the call? All right.
Yes,
Please
thank
go
you.
ahead with your question.
Thank you so much for that insightful presentation. It's a very amazing presentation. So my question is that phage's discovery has been around for a long time now, and there has been recent advances in its therapeutic invention and improvement in outcome of patient treatment. So, like, it seems to be a promising alternative to AMR, but my concern is that the resistance of even some strains of bacteria to some phages. What do you say to that? And how lasting is the solution of phages to antimicrobial resistance compared to antibiotics? Thank you. That's my question.
Very nice question. I hope I got it correct. So, regarding the bacteria resistant to bacteriophage, is it there? Yes, it is there. And I highlighted some of useful applications or synergistic effect of using phages either with antibiotics or with natural product to overcome this issue. And the simple way is to overcome the antimicrobial resistance is to isolate new phages and have what we call phage cocktail to control the resistance, especially that if we have like different phages that target different receptors. In addition to genetic engineering phages, which I personally believe in it, but I believe that using and isolating new phages is much more easier, especially in low and middle income countries like us. So simply, it is easier to find new bacteriophages and use phage cocktail. In comparison to antibiotic resistance, the phage resistant is simply can be handled by those solution, as I mentioned, by developing fish cocktail or use synergistic effect with antibiotics or with other natural product. While antibiotic resistant, again, antibiotic resistant is not that easy because mutations in bacterial strains cannot be challenged. Once it develops resistant to beta lactam or any other antibiotics, it is resistant, and simply it will multiply and produce billions of bacteria, which can spread over all over the world. So this is the advantage of having bacteria resistant to bacteriophage compared to bacteria resistant to antibiotics. I hope I answer your question, unless I missed something.
Thank you so much, sir. Kende, does that answer your question?
Yes, thank you very much.
Welcome.
All right. We have another person that wants to ask question. That's
Please.
Africa Phage Forum. Please go ahead with your question.
Yeah, hello. Can you hear me?
Yeah.
Yeah, good. This is Mutin. I'm just talking on behalf of the steering committee of Africa Phage Forum. I've seen a comment here that said Egypt should be a center for phage research in Africa. Yeah, sure. This comment, I would like Prof, since I know Prof is our mentor as well and advisor. We would like him to perhaps share with the community here present, because he's a busy man as well, some collaborative opportunity that they can get from his lab. Thank you.
Thank you very much, my colleague. And this is just a nice acknowledgement, but we all work together, and I will be more than happy to support anybody. And if APF is African Phage Forum is one of the most important forum, and I would like to thank them. I do collaborate with them as much as I can. And as he mentioned that I am quite busy, but I believe in this forum, and I believe to have the African Phage Society. We deserve it. We have many scientists in Africa region, Kenya, Egypt, Ghana, Nigeria, everywhere. So we have to join forces in the benefits of our countries. And thank you again. And I will be more than happy to help and support in any way. But please forgive me if I couldn't manage the time, because I have other administrative duties, which sometimes control my time. And I struggle with time management with many tasks at certain point. Thank you.
Thank you very much, Professor Ayman, for providing answers to all our questions. We really appreciate your presence. Thank you so much. Before we conclude, are there more questions, please? All right. Somebody is asking in the chat space if they can have your slides, sir.
This is a good question, which is difficult to answer. This is the only question I cannot answer clearly, because as you know, there are some patentable data included in this presentation, and some of them are not published yet, but at least if you are interested in any of our publication, I will be more than happy to share the original publication with you. But some of the data are not published yet, and they have, as I mentioned, some quality which is difficult to be shared right now. but I can share any publication out of my. Just contact me, and I will share it very, very easily. I will write my email in case if anyone interested to have any of my publication.
Thank you so much, sir. Um, I can see that Professor Ademo is also on the call with us. Thank you very much for joining, sir. Please, I'll give you the time to speak with us now. Thank you, sir.
Yes, sorry, I just want to say something. Can I go on?
Yes, please go ahead, sir.
Okay, thank you. I just want to use the opportunity to appreciate all the speakers of today. They've really made the program to be very, very rewarding. Many people, including myself, have learned a lot from this, and it is more addition to the existing knowledge that we have. So knowledge never ends. So we continue to gain more and more every time. So I really want to thank all the speakers, in particular the last two speakers, and also to thank and appreciate you, our students who have been able to package this. This is a yearly event, so we will continue to do it every year, and we believe that we will continue to have more opportunities, and we hope that we can have some collaboration both within Africa and across Africa and other continents. So it's just to appreciate everybody for all of this, and thank you all. So thank you very much. I am actually I am George Ademo. I am the head of the faculty that actually founded the Ibadan Bacteriophage Research Team, and this team is actually an undergraduate student affair. So we don't actually interject. We allow them to plan and organize everything. So the only thing I can do is only for me to just only come at the last minute like this and say thank you to everybody. So thank you very much.
Thank you so much, sir. We appreciate your presence. We really appreciate it. Thank you so much, sir. Thank you very much, Professor Ayman, for that eye-opening presentation and for providing answers to all of our questions. Since there are no other questions, we will. This is where we're going to end today's webinar. We're going to be continuing tomorrow. Yeah, the flyers are everywhere, so you can get the time to join the call again tomorrow. But before we conclude, finally, we will try to give chance for the display of the presentation from Nogin Biotech once more. Thank you. Nogin Biotech, we are waiting for the presentation. Okay, could you unmute your mic so that we can hear the sound too?
World's one and only phage DNA isolation kit at Norgem Biotech. Isolate high-quality DNA and support phage researchers worldwide. World's one and only phage DNA isolation kit at Norgem Biotech. Isolate high-quality DNA and support phage researchers worldwide. World's one and only phage DNA isolation kit at Norgem Biotech. Isolate high-quality DNA and support phage researchers worldwide. World's one and only phage DNA isolation kit at Norgem Biotech. Isolate high-quality DNA and support phage researchers worldwide. World's one and only phage DNA isolation kit at Norgem Biotech. Isolate high-quality DNA and support phage researchers worldwide.
All right, thank you very much, Norgem Biotech, for your presentation. Just to say this, we're going to be having the continuation of our webinar tomorrow, 25th October. We're going to be having Dr. Lavisha Parab and Sir Abokak Eli. The time is 10 a.m. to 11 a.m. UTC plus 1 and 11 a.m. to 12 p.m. UTC plus 1. Please make sure to be there so as to get more knowledge just like today. And we hope that we have a beautiful presentation as today again tomorrow. Thank you very much, everyone, for staying till the end. Really appreciate your presence. Thank you.